Good morning, friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order. You can make use of it. Thank you very much. So as we know, we are discussing about fetal growth and development. So today onwards, we will start with the development of a neurological system in the growing fetus, how it will going to develop. Now, after the formation of the trilaminar layers of the embryo, there will be subsequent development of the nervous system also, because all the development takes place hand by hand at a time. Okay, so we are discussing it one by one, but otherwise the development takes place simultaneously in all the systems, in the arterial system, in the musculoskeletal system, in the skeletal system, and also in the nervous system. Now, in the nervous system, there will be formation of the notochord and series of development takes place. The first thing is the formation of the notochord that we have seen when we are discussing about the trilaminar layers. So that itself is the base for the formation of the nervous system. Now the notochordal cells which are attached with the ectoderm, so which release certain special chemical substances upwards, and this makes the cells of the ectoderm to proliferate rapidly and produce an additional layer of the cells. Ectoderm is already there, which is adjacent to the notochord. Now, because of the release of certain special chemical substances by the ectoderm, now some of the new cells are formed and they proliferate and they make a separate layer of additional layer of the cells. That is, there will be thickening of the ectoderm. Ultimately, this results in thickening of the ectoderm because additional one layer is formed there. This thickening is now extend from the primitive node to the procaudal plate, and this extension is called as neural plate. Okay, so there will be thickening. This thickening actually start from the primitive node to the extend up to the procaudal plate, and this thickening is called as it is called as neural plate. Once the neural plate is formed, it start depressing the center downwards. That's development of a depression in the center point. It start depressing downwards, and later it start folding, and its edges now start raising up, and the center is now depressing further, and this is called as neural groove. So hence there is a formation of the neural groove. First there is a neural plate formed, then there is formation of the neural groove. The raised edges of this particular neural groove are called as neural fold. The folding will now continue till both ends come together and form a neural tube. Okay, that means the edges will continuously grow until they form a tube-like structure, and this tube-like structure is called as neural tube. So series of the changes taking place from the notochord. From the notochord there is a thickening, and then there is a formation of the neural plate, then there is a formation of the neural groove, then there is formation of the neural folds, and they now forms the neural tube. Now the anterior end is called as anterior end of this neural tube is called as the anterior pole, while the posterior end of this neural tube is called as the posterior pole. Okay. So this is how the anterior end and anterior pole and posterior pole is formed. This anterior pole becomes the future cranium, and the posterior pole becomes the caudal portion. Now some of the cells of the neural tube will not fuse. Okay, neural tube is formed, I told, but some of the cells of the neural tube has not still now fused. They are still open. The tube is not complete, remain separate, and it looks like a finger-like projections which are called as neural crest cells. Neural tubes are not completely fused at some points, and they have a finger-like projections, and these finger-like projections are called as neural crest cells. The anterior neural pole closes by the 25th day of the very important, this is very important, the anterior neural pole such a way formed, so that closes by the 25th day, while the posterior neural pole that closes by 27th day. Now improper closing of these poles, they create a lot of problems. This is the time the lady is usually missing her periods and expecting her pregnancy. 25th day, 27th day. Okay, so by that time, see, the neural tube formation is complete. Okay, but lady till now don't know that she is pregnant. So this is the time that lady is usually missing her periods and expecting the pregnancy. Around this period, lady should have sufficient amount of folic acid in her blood. That's the very important reason. Therefore, always we give more emphasis for uh, that means folic acid supplementation prior to the pregnancy. Okay, prior to the pregnancy, so we have to go for the supplementation of the folic acid, because mother is not knowing that she is pregnant by the time she is pregnant, and completion of the neural tube will take place. So all the female of reproductive age should always have folic acid in their blood, so that previously we have to supplement it. Otherwise, there will be some problem, because see, this is a neural. So if the proper closure of these poles will not take place, then there will be some permanent congenital problems will be there. Severe congenital problems will be there. That we will see later. Now, when the anterior pole and the posterior pole fuse, there should be folic acid in the blood, as I told. If it is deficient, then the anterior and posterior neuron pole may not close properly. This may lead to anencephaly. When the anterior pole not properly closed, that may lead to anencephaly, absence of the cortex, where rudimentary brainstem is formed, but upper part of the brain is not formed. So that is the anterior pole itself leads to the formation of the future CNS. And if it is not properly closed, for the proper closure, there should be large number of the cells should be produced. For the cells production, we need cell division. For that, we need folic acid. Okay. So now it is not properly formed, and only rudimentary brain is formed, and this condition is called as anencephaly. This baby cannot survive, born as dead baby, or dies immediately after birth or soon after birth. So anterior pole should be closed in time, for which the folic acid is very, very important. The posterior pole should also close properly by the time 27th day, and if not, the baby will develop different malformation in the lower back, caudal end, lower back, maybe spina bifida, meningocele, meningomyelocele, many things. So which are called as spina bifida and other group of anomalies. So after birth, in adult brain, the anterior pole is called as a lamina terminalis. Okay, so this is the basic thing. Now, what are the derivatives of the neural tube and neural crust cells? We know that what is meant by neural tube, what is mean by neural crust, the finger-like projection which is coming from the tube area. So that is called as neural crust. First, we'll see the derivatives of the neural tube. Neural tube has got cephalic end, we know that, anterior end, and also the caudal end, and above there will be precaudal plate. In the cephalic end or in the anterior end, the neural tube will forms the three ballooning, which will going to form the future brain. These are called as primary vesicles. These three ballooning like structures are called as primary vesicles. The proximal one is called as prosencephalon, which going to forms the forebrain. The middle one is called as mesencephalon, which will going to form the midbrain. The distal most is called as a rhomencephalon, which will going to form the hindbrain. In the middle part of the prosencephalon, it develops into two structures. The prosencephalon again divides into two structures. Out of them, one will grow very fast, and it is called as telencephalon, and another part which is very slowly growing, that is called as diencephalon. Okay, so now from the diencephalon we will have the development of thalamus, basal ganglia, pineal gland, subthalamus, etc. nucleus. Telencephalon, some portion of the telencephalon moves above the diencephalon and insula, diencephalon and insula, and develops into frontal, parietal, occipital, temporal lobe, which develops into cerebral hemisphere later. So this is how from the prosencephalon the whole brain is formed. Prosencephalon divided into telencephalon and diencephalon. From the telencephalon, basically the cortex is formed, cerebral hemisphere is formed, and from the diencephalon, basically thalamus and other nucleus will be formed. Rhomencephalon divides into two areas called as metaencephalon and mesencephalon. Metaencephalon and as well as mesencephalon. In the back, rhombencephalon. In the back of the metacephalon, a special plate develops which leads to cerebellum. There is formation of the cerebellum. Whatever left in the front is pons, and under it is called as medulla, and whatever left above is called as midbrain. Anterior pituitary will not develop from the neural plate. Now this is very important. Anterior pituitary gland will not develop from the neural plate. So it forms the Rothkes pouch in nasal mesoderm structures. Optic nerves will develop from the diencephalon. Now in the lower side, the neural tube develops and cells are called as neuroblast. They grows in four direction and are called as LR plate, two upper, and play a role in the sensory system, and the basal plate, lower one, so that play a role in the motor system. So from the motor cells, the fibers will come out and becomes the motor root. Similarly, the neural crushed cells, which are separated from the neural tube, will now start proliferating, and they will make a bulk and forms the ganglions. From this ganglion, some connection will come to the periphery, while some will go to the sensory side. These ganglions are called as dorsal nerve root ganglions, and which takes the sensation from the periphery and on the other side connected with the sensory gray matter. Some of the fibers from the sensory cells, different tracts are formed, which is running upwards, which makes the dorsal column, anterolateral column, etc. Now these are the derivatives of the neural crushed cells, melanocytes. Some of them are scattered around the whole neural tube and its derivatives like arachnoid matter, pia mater, which is totally called as leptomeninges. Dura mater is not a derivative of mesoderm. Sensory and autonomic, these are all the derivatives of neural crushed cells. Sensory and autonomic ganglions, dorsal nerve root ganglions, bones of the neurocranium, adrenal medulla. See, interesting, adrenal medulla. It is derived from the neural crushed cells. Basically, it is sympathetic ganglion. The visceral ganglia of the GIT, then calcitonin secreting parathyroid cells, odontoblast layer of the tooth, conotrunctal cushions, aorta septums of the heart, squam cells for myelination for the peripheral nerves, and these are all the development which takes place in the nervous system. So this is in total, in short, about the development of the nervous system from the growing embryo, in the growing embryo. Thank you very much. So we will meet in another class, and please don't forget to subscribe my channel, and also please give your valuable comments. Thank you.